Hi, I'm Michael Haney. I am a postdoctoral research fellow in Tony Wyss-Coray's lab at Stanford University. And today I'm really excited to talk about our lab's most recent research. But before I begin, I want to thank Oliver Cooper, Penny Howlett, and Ole Isaacson for giving us this opportunity to share our work. We're really happy to present at the Neuroregeneration Institute, part of the McLaren Hospital and Harvard Medical School.
The work we'll be talking about today is on some recent findings from our lab on the role of lipid droplet accumulating microglia in the aging brain and in neurodegenerative diseases. Much of the work we'll be talking about today was recently published in Nature Neuroscience in February of 2020, and this was work that was led by Julia Marschenlinger, a former postdoc in the lab. And towards the end of the talk, I'll mention some ongoing work that I'm leading, looking at this phenomenon in the context of Alzheimer's disease. So the story really starts with a chance finding that Julia made while looking at some electron microscopy of the aged mouse brain, where she found that within microglia, the resident immune cells of the brain, an accumulation of these lipid droplet structures. To a certain extent, these resembled the accumulation of lipid droplets in foamy macrophages involved in atherosclerosis, where these foamy macrophages are thought to play a very negative role in the disease, being senescent cells that secrete inflammatory cytokines and have increased reactive oxygen species. But what Julia noticed was that while these were abundant in the aged mouse brain, these were very rare or virtually absent in the young mouse microglia. To look at this in another way, by fluorescence microscopy, TMEM119 was used as a marker to specifically label microglia within the brain, and BODIPI was used as a dye to stain all neutral lipids. And what was seen in this context as well was an accumulation of these lipid droplets within the microglia of the aged brain while being virtually absent in the young brain. This is quantified here, where the number of lipid droplets in the per area of the dentate gyrus is significantly higher in the aged brain versus the young brain, and that a significant amount of the microglia within the aged brain have these lipid droplets compared to the young brain, and that even the size of the lipid droplets are much larger in the aged brain versus the young brain. The fact of these being lipid droplets was confirmed by staining with PLIM3, which is a protein that's known to associate with lipid droplets, and that this was within TMEM119 positive microglia. And with using a label-free approach by CARS imaging, which relies on the vibrational properties of molecules, it was seen that there are indeed lipid droplets within these TMEM119 positive microglia, and that these are abundant within the aged brain and much less within the young brain. To examine what types of lipids these lipid droplets are being composed of, lipidomics was performed on isolated lipid droplets from microglia in the aged brain. And what was noticed here was that while cholesterol esters compose many of the lipid, or is a big part of lipid droplets from other organs such as the liver, this was a very minor part of these lipid droplets, which were mostly composed of other phospholipids and glycerolipids such as TAGs and DAGs. So what's different about these microglia besides the fact that they have this lipid droplet accumulation? What other characteristics do they have? To address this question, the microglia were separated from the aged brain based on their lipid droplet content by FACS using this BODIPI stain. And RNA-seq was performed to compare the lipid droplet high versus lipid droplet low microglia within the aged brain. And what this showed was that the genes that were differentially expressed fell into a variety of categories, including phagosome maturation and reactive oxygen species production. To functionally investigate these, an in vivo phagocytosis assay was performed where labeled myelin was injected into the brain to see how the microglia cleared it based on their lipid droplet content. And what was noticed was that the lipid droplet high microglia had a selective deficit in phagocytosis compared to the lipid droplet low microglia. And this was also measured in a variety of assays in vitro as well. Additionally, primary microglia isolated from the aged brain was assayed for reactive oxygen species, and what was appreciated was that the lipid droplet high microglia had increased reactive oxygen species. And using a seahorse assay to look at metabolism, it was noticed that the NAD plus to NADH ratio in the lipid droplet high cells was much higher than the lipid droplet low cells or the young microglia, indicating a switch in metabolism away from oxytocin towards glycolysis. To examine the potential upstream regulators of this cellular state, the differential expressed genes were examined to see what other stimulators might produce this same type of transcriptional profile. And LPS was the top of the list, suggesting that a trigger of the innate immune system might be facilitating this process. Interestingly, APP was also on this list. But to investigate whether or not LPS is an inducer of lipid droplets, LPS was systemically delivered to young mice, and this did dramatically induce an increase in lipid droplets within the microglia of the young mice in the same way, to the same extent roughly that was observed in the aged mice. And looking at the transcriptome of these lipid droplet high cells in the young mice treated with LPS, they very much resembled that of the lipid droplet high cells in the aged mouse brain. Also, the lipid droplet high cells seem to be the ones secreting pro-inflammatory cytokines when treated with LPS. So this is most dramatic, perhaps, in the case of TNF-alpha. Another interesting inducer of lipid droplets that was noticed was aged human plasma. So in this instance, what we're showing here is that if you take a blood draw from an individual under the age of 25 and a blood draw from an individual over the age of 80, if you isolate the plasma, dialyze this plasma and delipidate it, and treat BB2 cells, which is a microglial cell line in culture with this plasma, the aged plasma selectively induces lipid droplets in these BB2 cells, and also induces this phagocytic defect that we've associated with these lipid droplet accumulating microglia. So one question we had is, if lipid droplet formation is blocked, do you prevent the associated dysfunctional cellular state, or are the lipid droplets simply a passive marker of this dysfunctional microglial state? To kind of tease that apart, there was a few tools at our disposal. Chief among them was trioxin C, which is a small molecule that blocks the ASCL family member proteins, which are necessary for lipid droplet formation. And what we noticed was that when you treat the cells with trioxin C, you prevent this LPS-induced lipid droplet formation in the BB2 cell line. It's the mouse microglial cell line. And when you block this lipid droplet formation, you rescue the phagocytic defect that is associated with the lipid droplet high cells. Additionally, you reduce the amount of reactive oxygen species that is produced in the lipid droplet high cells that are induced by LPS. What this data suggests, perhaps, is that the lipid droplet formation is upstream of the cellular dysfunction that we are seeing associated with these cells, perhaps. To get a broader look at the potential regulators of lipid droplet formation, we took an unbiased genetic screening approach. So we used the CRISPR-Cas9 system to do a genetic screen within these BB2 microglial cells. To do this, we expressed Cas9 in the BB2 cells, and we took a library of guide RNAs, roughly 30,000 guide RNAs, targeting 2,000 proteins with a bunch of negative control guide RNAs. And these 2,000 proteins are genes involved in proteostasis and cellular stress. And these guides were lentivirally infected into the BB2 cell line, such that each cell received one guide, making a single knockout. And these pooled knockout cells were then treated with LPS to induce lipid droplets. And then these were stained with a modified BODIPI. This is a phototoxic BODIPI that we got in collaboration with Carolyn Bertozzi's lab, that when a specific light is shined on it, it makes the BODIPI stained cells die. It's a phototoxic conversion. And so what we can do then is sequence the cells that remain after this selection and compare that to the cells at the beginning of this process just sequencing the guide RNAs to look at the change in the abundance, and this tells us which knockouts confer either block the lipid droplet formation or promote the lipid droplet formation. And what we saw from this genetic screen is that we got a handful of hits plotted here. So I'll describe this graph. On the x-axis is the effect size of the specific knockout. So a positive effect size indicates that this knockout inhibits lipid droplet formation, whereas a negative effect size indicates that this knockout promotes lipid droplet formation. And on the y-axis, we just have a p-value or a confidence score associated with these screen hits. And what we appreciated is that many of these screen hits are genes that have been previously associated with neurodegenerative diseases, such as progranulin, VPS35, which has been associated with Parkinson's disease, and MPC2, which has been associated with Niemann-Pick disease. So that was a very interesting finding for us. We went on to validate these hits individually by microscopy, so making knockout for these individual genes in the BV2 cell line and treating with LPS, and these validated very well. So for example, sorting exon 17, which is a gene from the screen that indicated that when knocked out, prevented lipid droplet formation. We see virtually no lipid droplets in this knockout, whereas progranulin, which is a hit which we found that when knocked out would promote lipid droplet formation, these microglial cells are chock full of lipid droplets after LPS treatment when compared to control. This is quantified down here, and importantly, the accompanying cellular dysfunction was either rescued or exacerbated in these knockout cells. So they either had more or less reactive oxygen species, depending on whether or not they made more or less lipid droplets, and the phagocytic phenotype was also modified in this fashion. Now, these were in a microglial cell line in culture using LPS. To look at this in a more realistic in vivo setting, the progranulin knockout mouse was used. So without any type of perturbation, we noticed that the microglia in the progranulin knockout mice had much more lipid droplets. This is in accordance to what we found in the screen data when compared to wild-type mice. And they also had the associated functional defects like decreased phagocytosis and increased reactive oxygen species. To kind of expand and follow up on these genetic screens, since that first targeted screen worked so well, I've recently conducted a genome-wide knockout screen for this phenomenon in a human monocyte cell line using this discovery of the old serum as an inducer of lipid droplets. So I've replaced the FBS in this cell culture model with either 10% human serum or 10% young serum, stained for the lipid droplets using the Bodipi dye, and then just simply used FACS to separate the lipid droplet high and lipid droplet low cells. And in doing this genome-wide screen, we actually got a lot more hits. We had roughly almost 500 statistically significant hits from this screen, regulating lipid droplet formation in the context of being induced by aged human serum. And the ones on the top of the list are all genes well known and well annotated to play a role in lipid droplet formation, such that the genes that when knocked out promote lipid droplet formation are the genes that are responsible for breaking lipid droplets down, so genes involved in lipolysis and lipophagy. And the genes that when knocked out inhibit lipid droplet formation are all genes known to play a role in lipid metabolism. And there's some interesting, we're investigating now some interesting novel hits further down the list that might indicate what role aged human plasma might play in this process. So to summarize the findings talked about so far, microglia in the aging brain seem to accumulate lipid droplets. These lipid droplet-rich microglia have this unique transcriptomic signature. We have dubbed this unique microglia state as LDAM for lipid droplet accumulating microglia. These LDAM have these distinctive functional defects such as decreased phagocytosis, increased reactive oxygen species production, and increased inflammatory cytokine release such as TNF-alpha. And we developed some novel CRISPR-Cas9 screens to discover some genes that are linked to neurodegeneration that play a role in this lipid droplet formation process. This includes progranulin, VPS35, sorting exon 17, and SLC33A1. So right now what we're wondering is, do these lipid droplet accumulating microglia, in addition to being a feature of aging, do they play a role in neurodegenerative diseases? And we were inspired to look into this actually by some of the original descriptions of Alzheimer's disease by Alois Alzheimer's himself. In his original publications describing the disease, he noted that many of the glial cells in his patients' brains have these large fat deposits. So we're intrigued by this. We're all very familiar with the plaques and tangles that he describes in these original observations, but very little is known or followed up on these fat deposits that he mentions. This was described again later in 1911, where the lipids themselves were specifically stained for. Just zooming in here, is noted that the glia cells stained positive for this lipid dye in these structures that very much resemble the lipid droplets that we're seeing, and that these are adjacent to the plaque cores in Alzheimer's disease. We've looked at this in some preliminary sense in the mouse models of AD. If you stain for A beta, you do see these lipid droplet accumulating microglia alongside of these plaques, and perhaps as well in human AD samples. But in addition to these early pathology descriptions, it's also been known in the human genetics community that there are genetic risk factors for Alzheimer's disease that are in lipid processing or lipid transporting genes. And this was highlighted in a recent meta-analysis of GWAS studies of Alzheimer's, where in addition to the well-known A beta and tau-related genetic risk factors, that there's this category of genes in lipid processing and lipid transport. They're now being appreciated as major risk factors for Alzheimer's. And just doing a simple gene ontology analysis of the GWAS hits, this is a very obvious and striking category of genes that might play a role in disease process. Perhaps the most famous amongst these and the most well known is the gene ApoE, which confers a huge risk for Alzheimer's disease. ApoE is a cholesterol transporter, or ApoE is a gene that encodes for a protein that is a cholesterol transporter. It comes in humans, it comes in 3 variants, known as ApoE2, ApoE3, and ApoE4, where ApoE3 confers is the most common and confers no risks, whereas Alzheimer's, whereas ApoE4 confers a stepwise risk in Alzheimer's, whereas having one variant of this, one ApoE4 allele gives you some disease risk, where being homozygous for ApoE44 confers significant disease risk. And interestingly, the other version, ApoE2, confers protection against Alzheimer's disease. And interestingly, this also is reflected in heart disease risk, perhaps due to its contribution to atherosclerosis risk. ApoE has actually recently gained a lot of attention in the context of microglia due to some newer studies using single-cell transcriptomics, where it's been appreciated that this microglial subtype known as DAM microglia, or disease-associated microglia, express very high levels of ApoE. And these might be related to Alzheimer's disease and in response to plaques. So they upregulate ApoE as well as other lipid-associated genes. And so this has really brought into focus the role that ApoE might be playing specifically within microglia in Alzheimer's disease. So the questions we had here are, do lipid droplet accumulating microglia play a role in a pathogenic role in Alzheimer's disease? And how does the ApoE genotype influence lipid droplet accumulating microglia? To address this question, we've formed this great collaboration with Thomas Beach of Banner Health, where it's been made available to us this excellent cohort of postmortem brain tissue with a large number of ApoE44 individuals that had Alzheimer's disease, which is rare to have this many samples, as well as Alzheimer's disease patients with ApoE33, so no genetic risk or protection against Alzheimer's, In addition, a cohort of controls with abnormally low pathology for their age. So all these are age and gender matched controls. So these individuals are individuals between the ages of 60 and 80 that have very low amounts of plaques and tangles, in addition to control individuals that have the normal amount of plaques and tangles associated with advanced age, but no cognitive deficits or other neurodegenerative disease diagnoses. So to study these in a kind of global, unbiased way, we performed single nucleus RNA sequencing. So I've isolated the nuclei from these frontal cortex samples, isolated the single nuclei using the microfluidic droplet-based capture from 10x genomics, and then performed RNA sequencing on these individual nuclei. In total, from all of these brains, we got roughly 170,000 nuclei to date, for which we have single cell transcriptomes, and we sequence these pretty deep at about average of 70,000 reads per nuclei. And if we plot all of these nuclei together on one dimensionality reduction, this is basically a PCA analysis, we can see that each of the nuclei cluster out into different cell types of the brain, with all the major cell types represented here and their percentages listed next to them. And so we're investigating the role that ApoE and Alzheimer's disease might play in all of these different cell types, but what I'll focus on are these microglia. We got a good number or good percentage of microglia from all these brains. And so what I did is I subclustered the microglia from all these individuals, and we could see that different clusters of different transcriptional states come out of this, suggesting distinct transcriptional states of different microglia. And if you overlay the sample origin onto these clusters, you can see that some of these are mostly defined, for example, from the control low pathology individuals, while others are dominated by the ApoE44 Alzheimer's disease individuals. In fact, this cluster up here, roughly half of the microglia from all of the ApoE44 Alzheimer's disease individuals is represented in this cluster. And if we look at what genes are defining this cluster, a lot of these genes are in processes that resemble the lipid droplet accumulating microglia that we saw from Julia's story. So these are genes involved in fatty acid biosynthesis, NAD metabolism, and TNF signaling. They're not the exact same genes that were observed in the mouse, but they're very similar in similar pathways, which is exciting to us. And just to highlight one of these genes, ASCL1, is dramatically upregulated in the ApoE44 AD microglia compared to controls. And so we're currently investigating this data set, and we want to intersect this with our previous CRISPR screens to find genes that are specifically upregulated in the disease state in different cell types, but that have a functional effect such that when they're knocked out, they decrease lipid droplet formation. ASCL1 is a well-known, well-characterized gene involved in lipid droplet formation. In fact, this is one of the targets of triaxin-C, this inhibitor that I was mentioning in Julia's study. But in addition, to this one, we're mining this data set now for new and novel genes that share this pattern, such that they were upregulated in the diseased brain and have a functional consequence when knocked out. And we're following up on those genes and different IPS models of microglia that are patient-derived, as well as looking back into the brains using CARS imaging. So in collaboration with Annika Enninger and Patrick Johnson here at Stanford in the Applied Physics Department, we're doing more CARS imaging, and these are on the same brains samples that we were doing the single-cell RNA-seq to confirm our findings there. And what's great about this setup is that we could stain in 3 different channels for different proteins. So here we're staining for A-beta and TMM119, the microglial marker. But in this label-free method, you can also image all of the lipids and even determine the types of lipids that are there. And we can image the lipids that might be inside microglia accumulating within these patient brains. And we're just getting started on this, and we're going to use this platform to validate our findings from the single-cell RNA-seq. But we're already seeing some indications that in the 80 patient brains, we're seeing accumulations of lipids within microglia, and this is a lot more rare in the control brains. But this is very preliminary and then ongoing work. So with that, I'd like to acknowledge all the individuals involved with this work. I'd like to acknowledge all the individuals in Tony Wyss-Coray's lab and Tony Wyss-Coray for his mentorship. Most of the work that I described here today was led by Julia Maschenlinger, a former postdoc in the lab. The single-cell RNA-seq work was done with the assistance of Nick and Kruti in the lab, and Christy and Rachel, the technicians, helping me out in the lab. We had some great collaborators that assisted in all aspects of this work and ongoing with Annika and Thomas Beach at Banner Health. And I'd just like to acknowledge our funding, the National Institute of Aging, and my particular funding in the Buck Institute of Aging. And with that, I'll wrap it up. Thank you for listening, and please feel free to reach out with any questions. Thanks.